Good afternoon, everyone. Today I would like to present some of my work on dengue virus host protein interactions that modulate infection in mosquito and human hosts. As we have heard from Leslie's talk, dengue is a flavivirus which is transmitted by the Aedes species of mosquitoes. Aedes aegypti and Aedes albopictus are the predominant species of mosquitoes that transmit the virus. It's estimated to infect around 400 million people every year, causing up to 25,000 deaths. Currently, there are no effective vaccines or mosquito control strategies that can limit the spread of the virus. Taking a closer look at the dengue genome, it's a single-stranded positive sense RNA virus. The open reading frame codes for 10 proteins, 3 structural and 7 non-structural proteins, and the genome is flanked by the 5' and the 3' untranslated regions, which play a very important role in regulating the viral life cycle. Once the virus enters the cell, the viral genome is released into the cytoplasm, where it undergoes translation to make the viral proteins, and these viral proteins help in replication of the virus, and the newly synthesized copies of RNA are assembled into virions and released. To complete each of these steps efficiently, the virus is heavily dependent on the host cellular machinery. So the dengue genomic RNA is known to interact with several host proteins that help it to co-opt the host metabolic machinery and evade the cellular immune defense mechanisms in both mosquito as well as human hosts. So what is very fascinating is that the virus is able to recruit host proteins from mosquitoes as well as humans, which are two physiologically and biochemically very different hosts. And we are interested in understanding how RNA protein interactions of the virus helps it to regulate its life cycle and helps it in adapting to both these hosts. In particular, we are interested in the 3' UTR of the virus, which is a hotspot for mutations. It's known to accumulate several adaptive mutations when the virus switches between mosquito and human hosts. It's also the site where the viral replication complex assembles, and the 3' UTR is known to be processed into several subgenomic RNA fragments, which are able to sequester away the host immune response proteins and protect the viral genome from the host immune defense mechanisms. And identifying these interactions and underlying mechanisms would be very important in developing effective antiviral interventions. The objective of our study is to identify the dengue 3' UTR host protein interactions and compare the RNA binding protein signatures in human as well as mosquito hosts. Further, we would like to test the functional role of these proteins in dengue host adaptation, immunomodulation, and pathogenesis. We have used a novel approach, a biotinylation-based approach called RAPID, to detect RNA protein interactions. This was recently developed by the Kavari lab at Stanford. RAPID is designed based on the highly specific interaction between the lambda phage box B RNA motif and the lambda N protein. So the RNA motif of interest, which in this case is the dengue 3' UTR, is cloned adjacent to the lambda box B RNA motif, and a biotin ligase is fused to the lambda N protein. Because of the highly specific interaction between the box B RNA motif and the lambda N protein, the biotin ligase is brought into close proximity to the RNA of interest. And when we add biotin to the medium, the biotin ligase is able to biotinylate the proteins that interact either directly or indirectly with the RNA of interest. So in our experiments, we co-transfect two plasmids, one expressing the box B tagged RNA and the other expressing the lambda N biotin ligase fusion protein into cells. And the biotinylated proteins can be captured using streptavidin beads, and biotinylated proteins can be identified by mass spec. For rapid experiments with the dengue 3' UTR, we cloned the full-length dengue 3' UTR as well as the 3' UTR derived sequence, shorter sequence into adjacent to box B sequences, and we co-express the box B tagged dengue 3' UTR RNA sequences with the biotin ligase lambda N fusion protein into mosquito embryonic C636 cells. Then we added biotin to the medium and captured the biotinylated proteins and identified them by mass spec. As a negative control, we used a scrambled RNA sequence, and we shortlisted proteins that were enriched in the dengue 3' UTR RNA expressing cells as compared to the scrambled controls. And proteins with high probability scores, which were highly enriched, were selected for further experiments. As you can see, there are several proteins that interact with both the full-length 3' UTR and the shorter version of the 3' UTR. We identified around 40 proteins that could possibly interact with the dengue 3' UTR either directly or indirectly as a part of a complex. And most of these proteins were RNA binding proteins and ER associated, endoplasmic reticulum associated proteins. We selected a few proteins that could bind to both the longer and the shorter versions of the 3' UTR for further experiments. So these are the proteins that we shortlisted, and these are the corresponding human homologs. In the next step, we decided to test the effect of knockdown of these proteins on dengue replication in mosquito cells. We used double-stranded RNAs around 500 base pairs in size to knock down specific proteins in mosquito cells. And after knocking down specific proteins, we transfected these cells with, we infected these cells with dengue. And 4 days post-infection, we harvested the cells for quantifying the intracellular viral RNA levels by quantitative PCR. So this panel shows that the proteins that we wanted to knock down have been efficiently knocked down. And amongst the proteins we tested, we noticed that knockdown of Sec61A1 and loquacious proteins leads to a significant decrease in the dengue RNA abundance in mosquito cells. So what are these proteins? Sec61A1 is an endoplasmic reticulum associated protein that is involved in protein transport across the ER membrane. Recently, it was shown that small molecule inhibitors of Sec61A1 can effectively reduce the dengue viral RNA loads in both human and mosquito cells, which makes it a very good positive control for our assay. The other protein that we saw which had a significant effect on dengue was loquacious. Loquacious is a double-stranded RNA binding protein. It is known to interact with dicer proteins as a part of the RISC complex and modulate siRNA and microRNA biogenesis in insect cells. And we are very interested in learning more about its role in the viral life cycle. So as you can see, when we knock down either of these proteins, Sec61A1 or loquacious, there is a significant decrease in the dengue RNA levels as compared to the negative control. There is also a significant decrease in the viral protein levels, indicating that both these proteins are proviral, that is, they are essential for the virus to replicate efficiently in mosquito cells. We also tested if these proteins are required by other flaviviruses. So when we knock down either of these proteins, we could see that there is a significant decrease in the viral RNA abundance for most of the flaviviruses, dengue, yellow fever, as well as Zika virus. We did not see any effect on unrelated alpha virus, which is chikungunya virus, indicating that these proteins are essential for the replication of other flaviviruses as well in mosquito cells. So far we have seen what happens in mosquito cells, and now we were interested in looking at the human homologs of loquacious. So there are two human paralogs of loquacious, TRBP and PACT. As you can see, the RNA recognition motifs in these 3 proteins are very well conserved. They're up to 60% identical. And similar to the role of loquacious, even PACT is involved in in interacting with dicer and modulating the RISC complex activity in human cells. So what happens when we knock down PACT? Does it also have a similar effect as we see with loquacious? So first we knocked down one of the paralogs, which is TRBP, using siRNAs, and we did not see any effect on dengue replication. However, when we used a PACT CRISPR knockout, we could see that there is a significant reduction in the viral RNA levels, indicating that PACT is essential for dengue replication in human HUH7 cells. And these observations lead to a lot of exciting questions that need to be answered. How do loquacious and PACT regulate the dengue life cycle? Are the mechanisms similar in the 2 species, or are they different? Can the phenotype be rescued by overexpression of the corresponding proteins? And can this phenotype be also seen in live infected mosquitoes? These are the questions that we are really excited to answer in the future. And to summarize what you have heard so far, we adapted RAPID to identify RNA protein interactions of dengue 3' UTR and mosquito cells. And we identified 2 important proviral factors, Sec61A1 and loquacious, in mosquito cells. And interestingly, the human paralog of loquacious, which is PACT, also showed a similar proviral phenotype, where it was essential for dengue replication in human cells. So I would like to acknowledge the Sarno lab members and Current lab members, particularly Yoshin, who did viral infections with different flaviviruses. I would like to thank the Stanford University mass spec facility, where we did all our mass spec experiments. I would also like to acknowledge the valuable inputs by Andrea Gamarnik, our collaborator, with whom we are trying to design viruses, infectious viruses which have the box B tag. And Mariano from UTMB, with whom we are doing infections in live mosquitoes and knocking down loquacious and trying to look at the phenotype in vivo. I would like to thank all of you for your attention, and I'd be happy to take questions.
Thank you. That was really exciting. I guess maybe you haven't tested yet, but do you know if PACT and loquacious go to the replication complex? And also, could you harbor a guess? What do you think loquacious and PACT are doing in a proviral manner?
So we have not really tested whether they are actually present in the replication complex, but we do know that both of these proteins act in the very early stages of viral infection at very early time points. So they are either involved in modulating translation or replication of the virus. So there's a good possibility that they could be a part of the viral replication complex. And as to the possible role that they could be playing, with loquacious, at least, we think that it might be, there are 2 possibilities. One is because it's a part of the RISC complex, it's possible that it's modulating siRNA or microRNA biogenesis in the cells and indirectly affecting the virus. The other simpler possibility is that by directly binding to the viral RNA, it's able to recruit other host proteins that are required.
So Sec61 was identified in a protein screen and in the RNA binding screen. I mean, the previous paper was showing that it was also identified as a protein binding factor, if I recall it correctly. So you think there is what's the connection between the viral proteins and the RNA here with Sec61? You think that it's a viral RNA, that the viral protein that binds RNA binds then the Sec61, or do you think that's just 2 different things?
So the first thing is, because we have used just the dengue 3' UTR in our experiments, the system does not have any viral proteins. So Sec61 is probably not being recruited through its interaction with viral proteins. And some recent studies have shown that it has non-canonical RNA binding activity, the Sec61 protein, through which it can possibly recruit RNAs to the endoplasmic reticulum. So we suspect that its binding to the viral RNA could be recruiting the viral RNA to the ER.
Just a quick question. I guess this will get really interesting looking at tissue expression of these proteins and whether some are redundant in function or not.
Yeah. So, loquacious has been seen to be expressed in all tissues of the mosquito. They have checked midgut and salivary glands and so on. But there's a paralog of loquacious called LOX2, which is specifically absent from the midgut. So it's possible that there is some interplay between these two paralogs, which would be interesting to look at in different tissues.
Thank you. And this, another question?
To what do the adaptive mutations affect the binding of either of the proteins in recovery?
We haven't tested those yet, but we are planning to use both the mutant and the wild type in our rapid screen and look at differential binding.